Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 10th February 2021. As you know, we are into the revision mode, and this time around we are revising the entire syllabus in the form of sample papers, and already we have answered two sample papers, and this is the third sample paper. And in this we have answered up to section B. Now today in this session we'll go to section C. Now I'll read the questions followed by the answer, and as I said, you need to keep a separate notebook for these 10 to 12 sample papers that I'm going to give, so that it will be very handy during your examination. Section C, question 26. And section C, you know that there will be 5 questions, 3 marks each, 5 into 3, 15 marks. So question 26th, work out a cross between true breeding red and white flowered dog flower plants, that is snapdragon, up to F2 progeny. Explain the results of F1 and F2 generation. Answer, in F1 generation, pink flowered plants are obtained. It is due to incomplete dominance. Now you need to you put the checkerboard. In F2 generation, alleles of the hybrid segregate during gamete formation, and the parental characters reappear without any change. So the phenotypic and genotypic ratios of F2 generation are same. That is, capital R small r is to capital R small r is to small r is 1:2:1. What is the normal ratio? It should be 3:1. So there is a deviation. Instead of 3:1, it is 1:2:1 because of incomplete dominance. And you can also explain this answer by putting a cross, okay? Monohybrid cross. Question 27. The total number of genes in humans is far less, that is less than 25,000, than the previous estimate up to 1,40,000 genes. Comment upon this. When scientists began estimating the number of human genes, they began with a very high figure, that is more than 1 lakh. At that time, the technology for studying human genes was not sophisticated enough, and the estimate was more qualitative in nature, as it was mainly based on assumptions. With the gradual progress of technology and knowledge about human genes, the estimated number began to come down. The present knowledge tells us that the total number of genes in humans is between 20,000 to 25,000. Question 28. What precautions would you recommend to a patient requiring repeated blood transfusion? Answer, repeated blood transfusion may result in contracting diseases like AIDS. The recipient must ensure that the donor's blood is being screened for HIV and other pathogens. Also, he should make sure that doctors are using fresh needles. Question 2. If the advice is not followed by the patient, there is an apprehension that the patient might contract a disease that would destroy the immune system of his or her body. Explain with the help of schematic diagram only how the immune system would get affected and destroyed. So we have said what are the precautions that one must take. In the absence of such measures, the patient can get infected by HIV, human immunodeficiency virus, which causes AIDS. It's a threatening disorder that weakens the immune system by attacking helper T cells in the body. A schematic diagram showing a cycle of proliferation and effects of HIV in infected person is as follows. Now you got a retroviruses and viral RNA core, viral protein coat. And the virus infects the normal cell. This is the normal cell, and in the normal cell it's entering inside, okay, through plasma membrane. The viral RNA is introduced into the cell. Viral RNA is produced by reverse transcriptase. Now, once that happens, viral DNA incorporates into the host genome. So, there is a duplication of RNA plus RNA becoming DNA. That viral DNA gets incorporated into human DNA. So, new viral DNA is produced by the infected cell. The new viruses, once this gets broken, the human DNA, which is incorporated with the viral DNA, then you have got new viruses that are produced within the human cell. So as a result, the human cell gets blasted off, and new virus can infect other cells and decrease the number of helper T cells. So immunity of the person thus gets deficient or decreased. Now question number 29. Differentiate between the process of transcription in prokaryotes and eukaryotes. Transcription in prokaryotes and transcription in eukaryotes. Products of transcription become effective in situ. Products of transcription come out of the nucleus for functioning in cytoplasm. There is only one RNA polymerase in prokaryotes. 3 RNA polymerases take part in it. mRNA is polycistronic, whereas in eukaryotes, mRNA is monocistronic. Splicing is not required in prokaryotes, whereas splicing is required for removing introns. Now question number 30. Mention the name of causal organism, symptoms and the mode of transmission of the disease amoebiasis. The causal organism, of course, all of us know, is Entamoeba histolytica. So take care of the water that you drink. And symptoms: severe abdominal pain with cramps, stool with blood and mucus. Mode of transmission: the tetranucleate cysts of the parasite transmitted through contaminated food and water. That's why one must take very hygienic food and clean water. Now there is a choice for this question. What is the basic principle of vaccination? How do vaccines prevent microbial infections? Name the organism from which hepatitis B vaccine is produced. So nowadays, of course, this word is very, very familiar and common. Vaccine, like corona, COVID-19 vaccine. So the principle of vaccination is based on the property of memory of the immune system. In vaccination, a preparation of antigenic proteins of pathogens are inactivated or live, but weakened pathogens are introduced into the body. The antigens generate the primary immune response by producing antibodies. The vaccines also generate memory B cells as well as T cells. When the vaccinated person is attacked by the same pathogen, the existing memory B cells and T cells recognize the antigen quickly and fight the invaders with massive production of lymphocytes and antibodies. Hepatitis B vaccine is produced from, as we said yesterday, from yeast. Now we go to section D. Section D, as I said, it is a long answer type questions, 5 marks. 3 questions will be there, again 3 into 5, 15. So section C and section D have got equal importance, 15 marks. Now question 31. Explain the process of spermatogenesis in humans. Spermatogenesis is the production of sperms in males. In testes, the immature male germ cells, spermatogonia, produce sperms by spermatogenesis that begins at puberty due to significant increase in the section of gonadotropins, that is luteinizing hormone and follicle stimulating hormone, under the influence of gonadotropin releasing hormone GnRH released from hypothalamus. B. Spermatogonia, singular spermatogonium, present on the inside of wall of the seminiferous tubules, multiply by mitotic division and increase its numbers. Each spermatogonium is diploid and contains 46 chromosomes. Some of the spermatogonia, called primary spermatocytes, periodically undergo meiosis. The primary spermatocyte undergoes meiosis 1 and forms two haploid secondary spermatocytes containing 23 chromosomes each. That means 2n has become n. The secondary spermatocyte undergoes meiotic 2 and form four equal-sized haploid spermatids. Spermatids now transform into spermatozoa by spermiogenesis. Now see, spermatogenesis is different, spermiogenesis is different. After spermiogenesis, the sperm heads are embedded in the serotoli cells and released from the seminiferous tubules via spermiation process. Now three are there: spermatogenesis, spermiogenesis, spermiation. Now question 2, draw human sperm and label acrosome, middle piece. Mention their function. Now this is acrosome and this is the middle piece. Now anyway, since you are drawing it, label all. And then the function of acrosome. Acrosome is filled with enzymes that help in sperm to penetrate the ovum. So it's very, very important having enzymes. And middle piece possesses many mitochondria to produce energy for the movement of the tail to facilitate sperm motility. Or choice is there. Figure question 1. The diagrams identify the figure that illustrates corpus luteum and name the pituitary hormone that influences its formation. Now corpus luteum, figure G, of course he has given almost up to I, A to I. Figure G illustrates corpus luteum and it is luteinizing hormone LH. Question 2. Specify the endocrine function of corpus luteum. How does it influence the uterus? Why is it essential? Corpus luteum secretes progesterone hormone, which stimulates the continued growth of superficial layer of endometrium, and endometrium becomes ready for implantation. In anticipation, it gets prepared. It is essential for continuation of pregnancy. Question 3. What is the difference between D and E? The figure D represents a fertility follicle in which primary oocyte completes its first meiotic division, while figure E represents the mature follicle, that is Graafian follicle, ready for ovulation. Now question 4. Draw neat labeled sketch of Graafian follicle. So you need to draw the sketch like this and label it. Theca, granulosa cells, antrum, cumulus ophras, zona pellucida, secondary oocyte. So this is how you need to draw the diagram. Now question 32. Given below is a table depicting different restriction enzymes and its examples. Restriction enzymes A, B, example SMAL and EcoR1. Now name the following type of restriction enzymes A and B. The type of restriction enzymes A is restriction endonuclease. B is also restriction endonuclease. Both are endonucleases. Question 2. What is PCR? For what purpose it is used in biotechnology? Draw well-labeled diagram showing different steps in PCR. PCR stands for polymerase chain reaction. This technique is used for gene amplification, making the gene of interest into more numbers. The well-labeled diagram showing different steps of PCR is follows. Now this is reason, red color one is reason to be amplified. Of course you got 5' and 3' ends and 3' to 5' double-stranded DNA. So first one is denaturation. How that is done? Heating. And then after that annealing. So you have taken a primer and you are extending that primer on both the sides. And then third step is extension. Once the annealing is done, you are extending the primers. So this is how number of cycles it will be repeated so that you get around 1 billion times amplified one. That is the use of it. Okay, 30 cycles. Now PCR, each cycle has 3 steps. Primer annealing and extension of primers. Now question, there is a choice. Why must a cell be made competent in biotechnology experiments? How does calcium ion help in doing so? Since the DNA molecule is hydrophilic, they cannot pass through cell membranes. For recombinant DNA to be integrated into the vector or host genome, it is necessary for the DNA to be inserted into the cell. Therefore, making the host cells competent is necessary for biotechnology experiments. The two ways by which cells can be made competent to take up DNA are chemical action. A. The host cell is treated with specific concentration of divalent cation, that is calcium, which increases the pore size in the cell membrane. DNA is then incubated with the treated bacterial cell at 40 degrees centigrade, thereby increasing the efficiency of DNA entering through pores in the cell wall. Second one, of course, very familiar one, heat shock treatment, incubating the cells with recombinant DNA on ice, followed by brief treatment of heat at 42 degrees centigrade, and again putting them back on ice. Second one, state the role of ballistic gun in biotechnology experiments. Ballistic guns or gene guns, they are commonly known as, are used to bombard rDNA loaded on gold and tungsten particles with high velocity. In this way, the rDNA is delivered to the desired host cells. Question 33, last question. Name the category of microbes naturally occurring in sewage and making it less polluted during the treatment. The category of microbes naturally occurring in sewage and making it less polluted are two, bacteria and fungi, wherein masses of bacteria get associated with the filaments of fungi to form mesh-like structures called flux. Question 2. Explain the different steps involved in the secondary treatment of sewage. The different steps involved in secondary or biological treatment of sewage is, the secondary treatment of sewage is also called biological treatment because in this treatment, sewage is subjected to biodegradation. It means that it involves the participation of microbes. The process of secondary treatment involves the following steps. A. Primary effluent is passed into large aeration tanks with constant mechanical agitation and air supply. This allows vigorous growth of useful aerobic microbes into flocks, mass of bacteria and fungal filaments. B. These microbes consume a major part of organic matter in the effluent while growing. This reduces the BOD of effluent, biological oxygen demand. C. When BOD of sewage gets reduced, it is passed into the settling tank. The bacterial flux settle in the tank and the sediment is called activated sludge. A small amount of activated sludge is pumped back into the aeration tank to serve as inoculum. D. The remaining major part of the sludge is pumped into large tanks called anaerobic sludge digesters, where other kinds of bacteria which grow anaerobically digest the bacteria and fungi in the sludge. During this process, bacteria produce a mixture of gases such as methane, hydrogen sulfide, and carbon dioxide, which form biogas. So, what are those? Methane, hydrogen sulfide, and carbon dioxide. The effluent from secondary treatment is generally released into the natural water bodies. It helps to reduce water pollution and waterborne diseases. Now there is a choice for this last question also. Secondary treatment of the sewage is also called as biological treatment. Justify the statement and explain the process. Again same, it is almost a repetition. The secondary treatment of sewage is also called biological treatment because in this treatment, sewage is subjected to biodegradation. It means that it involves participation of microbes. The process of secondary treatment involves following steps. Primary effluent is passed into large aeration tanks with a constant mechanical agitation and air supply. This allows vigorous growth of useful aerobic microbes into flocks, mass of bacteria and fungi. These microbes consume a major part of the organic matter in an effluent while growing. This reduces BOD of the effluent. When BOD of the CVS gets reduced, it is passed into settling tank. The bacterial flocks settle the tank and sediment is called activated sludge. A small amount of activated sludge is pumped back into aeration tank to serve as inoculum. Just like when your mummy is preparing curds, she puts a small sample of curds into the milk. That's inoculum. The remaining major part of the sludge is pumped into large tanks called anaerobic sludge digesters, where the other kinds of bacteria that grow anaerobically digest true bacteria and fungi in the sludge. During this process, bacteria produce a mixture of gases such as methane, hydrogen sulfide, and carbon dioxide that forms biogas. So the effluent from secondary treatment is generally released into natural water bodies. It helps to reduce water pollution and waterborne diseases. So this is how you need to answer large type of questions. That is, point wise you have to answer.